For the introduction, Tyler, and thanks everyone for coming to listen to my talk. So today I'm going to talk about water circulation and water quality dynamics in here fish pond during restoration regimes. And I'm doing this research at the Department of Oceanography at the University of Hawaii in Mānoa. And on this picture you can see here fish pond, which is located at the terminus of here watershed on the windward side of Oahu in Kaneohe Bay. And before I get started talking about my research, I really want to acknowledge all the people that have greatly contributed to this research. So my advisors, Rosie Allegado and Margaret McManus, and then my research committee, Brian Glazer and Craig Nelson, and all the people in the Allegado lab that have really greatly helped with field work and lab work, and also my funding sources, the Department of Health and Sea Grant, and Paipa'uhia, who are the community students of here Fish Pond that have always been really welcoming to researchers and in informing us about their work at the fish pond. So this is really a very collaborative effort. So first, I'm going to talk a little bit about the background and conservation ecology, which is kind of the larger picture where my research fits into. I'm also going to talk about the resilience of native Hawaiian systems and here fish pond as an example of restoration. And then I'm going into my two major research questions: what is the effect of restoration on water circulation? And also, what is the effect of circulation change on water quality? And this is measured by looking at microbial communities in the water. So Hawaii has been, as many of you probably know, has been facing a variety of challenges with global warming and land use change. So some examples of these are beach erosion, which you can see on the picture on the top left. I'm sure you've seen pictures like that. The top right shows the recent flooding on Kauai, which probably wouldn't have been so bad if the land wouldn't have been changed so much in the past century. The top see an example of plastic pollution because Hawaii is located in the middle of the subtropical gyre. We are very prone to accumulating plastic waste on our beaches. And in the bottom right, you can see, this is a picture that was taken from Diamond Head very recently after one of those major rain events. And you can see how the water is really discolored and almost brown. We call it brown water due to a lot of sediment erosion from those rain events. And that also impacts water quality greatly. And so conservation ecology really looks at, it's a very interdisciplinary effort to look at how we can maintain the integrity of these ocean ecosystems in the face of rapid global change and anthropogenic impact. And so questions in conservation ecology are how can we foster resilience of ecosystems, or also how can we manage invasive species on an ecosystem scale? Or what impact does restoration have on the different parts of the ecosystems? And this is really where my project ties into. So I'm looking at the impact of restoration on here fish pond. So fish ponds are one of the most ancient and advanced forms of aquaculture. You can see here on the top right, they are part of the traditional ahupua'a system, which is the Hawaiian word for watershed. And this is really a very holistic form of land use management. And you can see usually they have these flooded agroecosystems further upstream, often based on taro, that would capture a lot of the sediment runoff and water. And so the water would circulate in these taro fields that are also called lo'i, and then accumulate a lot of nutrients in this process. and flow further downstream. And then usually we had the fish ponds at the terminus of the stream that would very much benefit from this nutrient-rich water. And so two of the target species in these Hawaiian fish ponds are limu, which is a form of macroalgae that you can see here on the bottom right, and ama, which is a striped mullet, a herbivoric fish that was mostly feeding on these limu. And so this combination of nutrient-rich water from Hia Stream, but also the influence of salt water from in this case, Kaneohe Bay, and shallow water conditions and a lot of sunlight radiation really caused these amazingly productive estuarine-like conditions that fostered the growth of algae and then attracted a lot of herbivoric fish that then fed on the algae. So from an energy efficiency point of view, because only 10% of food material is incorporated into the next trophic level, this system was really efficient because you could feed a lot more people with herbivoric fish than, let's say, fish like tuna, apex predators, which are much higher up in the food web. So yeah, so this very holistic system, and the Hawaiians very much understood that all these elements in the ahupua'a are interconnected. This system really reflects on the deep understanding Hawaiians had on the environment specific to these islands. And they really cultured and protected these systems. So here, fish pond is a local kuapā type of fish pond, which means that it is completely encircled by a wall, a man-made constructed wall made from fossilized corals and basalt. And it has these sluice gates that foster water exchange in the wall that are called mākāhā. And so because of this enclosed nature, the fish pond is really an ideal model system to study the coastal environment because it allows for a degree of experimental control that wouldn't be possible in an unconfined system. So mākāhā are these wooden sluice gates. Here on the left, you can see a depiction of the structure of one of these Hawaiian fish ponds with an integrated mākāhā. So usually they are these wooden grids that help recruitment of fish. So usually how it works is that small fish are attracted by the algae inside in the nutrient-rich conditions. And so they fit through these mākāhā grids and then grow very quickly in the pond until they are too large to be able to swim back out. So this is really a very cleverly engineered system. And it also allowed the fish harvest because at low tide, when the water was flowing back out, a lot of fish would just accumulate in these mākāhā channels. So the Hawaiians would usually open one of these grids, and then the fish would swim in, but they couldn't exit into the ocean, and they would close the second grid and then had a lot of fish just in this mākāhā channel that was basically ready to be harvested. So, and another important function of these mākāhā was really mixing and water exchange. Because it is such a shallow water environment, it is important, fish is so dependent on stable oxygen levels and good water quality. It was important to have enough circulation and change. And also, of course, the amount of mākāhās defined then the residence time, because the more mākāhās you had, the more exchange you had, and the shorter the residence time. The residence time is the time that a water volume spends in a place before it is exchanged. The kuapā, the wall, had several functions. First of all, it contained the water during very low tide, so it really prevented the fish pond from completely draining. So they ensured that you would always have a minimum water level for the fish to survive. And they kept the target fish inside while protecting the fish from larger predators outside. So in the last century or so, due to a combination of social, economic, and natural impacts, we unfortunately went from a really well-functioning integrated system to a system very low in productivity. And some of the reasons are that we changed the land use. So we went from this traditional ahupua'a system based on the flooded agroecosystems to a more plantation-style agriculture system that was much less efficient in capturing the sediment. And this is really reflected on these two pictures. You can see from the left one from 1928 and the right one from 1959. You can see just by the change in color in the pond how much sediment was really discharged into the pond interior. Another problem is urbanization, and invasive species is also has been a problem in Hawaiian fish ponds as well. So I'm going to talk a little bit of invasive species. Mostly here I'm talking about mangrove. In many parts, if they are native in many parts of the world, they are really appreciated for the ecosystem services that they provide. So some examples of that are shoreline protection, sediment stabilization, litter fall subsidy, or the provision of nursery grounds for fish. But when not, and on the picture on the left, you can see a very healthy ecosystem. But in Hawaii, they were introduced intentionally to capture some of the sediment runoff. And then they caused basically a cascade of them, being that they basically transformed the natural sandy habitat into a very heavily vegetated area that inhibited water exchange and water flux, and also accumulate sediments. And also the mangrove litter caused large-scale anoxic sediments. In an environment like here, fish pond, which is really shallow and constant water exchange and stable oxygen levels, this is really emissions. And so the removal of mangroves is one of the major management practices of fish pond. So OHIA, which is a non-profit organization that really aims to restore the fish pond to its non-impacted state, started their work basically like this. So all of the river mouth and all the pond periphery, including the wall, was overgrown by thick mangrove forest. And this was back in 2003. So this is how the fish pond looks now. You can see that they made really amazing progress in removing the mangroves around the pond periphery. And sometimes you can see that these mangroves, they leave behind these areas of really thick sediment accumulation. And on the graph on the right, you can see really the chronos sequence of mangrove removal over the past 10 years or so. And you can see that the blue areas indicate where mangroves were and where they have been completely removed. And only the red line shows where there's still mangrove forest. And the ultimate goal is really to remove all the mangroves and clear the area completely from this type of invasive species. And so removing the mangroves was really important also to be able to restore parts of the wall that were broken during a major flood in 1965. So there was this large section, on the top right, you can see this large section that was broken during the flood. It's about 50 meters long, and it was flooded during high tide. And so in 2015, with a big community effort and a lot of community work days, they managed to restore this part of the wall completely, and they integrated a new mākāhā channel also into the wall that they named Kahokela. And the section that was broken, because I'm going to refer to this later on as well, before the repair was called Ocean Break. So these are just some pictures of the restoration efforts that have been going on. So you can see they have a lot of community work days, I believe every second and fourth Saturday of each month. You can come and help Paipai'ohia with their restoration work. And yeah, you can see people working on cutting down and burning mangroves, transporting rocks through water to repair and restore the wall. And yeah, this really reflects on the great community effort that has been going on over the last years. So now that I talked a little bit about the background, I want to come to my first hypothesis, basically. So we are basically looking at what is the effect of restoration on water circulation. And while the restoration effect probably affected a lot of areas in the fish pond, here we are focusing basically on the impact that the repair of Ocean Break and the addition of a new mākāhā channel had on water circulation. And we hypothesized that Ocean Break repair affected the dominant flow pathways for water exchange. It also affected likely fish pond volume and residence time. And we also think that the salinity distribution, because these two water masses that influence the geochemistry at Hia Fish Pond, the fresh water from Hia Stream and the salt water from Kaneohe Bay, really reflects or can be used as an indicator for fish pond circulation dynamics in the interior. And we hypothesized that because we added a new mākāhā channel in, that the fish pond exchange increased due to the repair of Ocean Break. So some of the questions we wanted to answer are: how fast is water flowing in and out the fish pond at different tides? How much does each mākāhā contribute to water exchange? And what is the fish pond bathymetry and associated volume? So how did we approach this? We deployed current meters at each of the mākāhā because we really wanted to quantify flux going in and out of the fish pond. And these current meters measure current velocity, direction, and water level. And this is a little video. Oh, probably need to. Just giving you an idea of how fast the water is flowing and how one of these mākāhā channels looks like. So there's 6 mākāhā channels that we measured water flow, and we deployed these current meters here. These are just some pictures of the instrumentation we used. So in the center, this is a Nortec Aquadrop flow meter. And on the right hand, you can see basically a flow meter that measures the same thing. So velocity and current direction. And in the beginning, we deployed both of them to see which one works better, because the conditions are very shallow and very channelized flow, and not all current meters work very well in these conditions. And so we ended up using the one on the right, the Sontec, that was then powered by solar batteries. And on the left, you can see one of those pressure sensors that we use to monitor water level. Here's just some pictures of the field work. So on the left, you can see, it's a little bit hard to see, but there's one of the Sontecs deployed and then one of the Nortecs. So we deployed both current meters at the same time to figure out which one gives more accurate data in these conditions. And because the fish pond is so shallow, we had to It's possible to walk around on the wall, but because the instrumentation is so heavy, we often, when it wasn't possible to take the boat, we used a kayak to load all the gear on it and then kayak to the different sites. So we didn't have to carry the gear all around. So these are some pictures of the field work we did out there. And now I'm going to show you some of our data. So this is the water pressure data that we collected. And we collected data over several tidal cycles. And this is really important because, as you can see, the tidal amplitude really changes a lot over time. So in Hawaii, we have mixed semi-diurnal tides, which means that we have 2 high tides and 2 low tides in 24 hours, but they are not all the same height. And so you can see that we have these larger peaks and smaller peaks. And then you can also see that the tidal amplitude changes over the course of a week greatly. And that's because we have something called spring tide and neap tide. And spring tide is basically when the moon and the sun align. And so the gravity pull is much stronger and leads to much higher tidal amplitudes. And during neap tide, we have smaller tidal amplitudes because the sun and the moon are basically in a right angle. And so they pull into different directions and cause a smaller tidal amplitude. And so because the tide varies so much, we wanted really to collect different tidal cycles and capture the data from that. And then we also really wanted to establish rating curves that relate water volume flux to the water level. And this was really important to us because pressure sensors are so much smaller and easier to handle and so much cheaper than those large current meters. And so once we establish a relationship between water volume flux and water level for each of these different tidal states, we can just deploy these small pressure sensors over long term to then basically get water volume flux and make an estimation of the water coming in and out just based on the pressure. So that was the whole idea of the rating curves. So here you can see an example of these rating curves. Water volume flux is determined using this equation down here. So it's basically just the mākāhā dimensions multiplied with the water level that changes over time. And pressure, I forgot to mention that earlier. Pressure is really an indicator for water level. So you can translate pressure one-to-one into water level or water height. And that is then multiplied with water velocity to receive water volume flux, which is on the x-axis of these graphs. And then the water level is on the y-axis. And this is the relationship between water level and water volume flux. And you can see that as the tide rises, so the water volume flux increases and is maximal during mid-high tide. And then it decreases again until the flood tide switches into ebb tide. And then the water is basically flowing out of the fish pond. So then the water volume flux becomes negative. And on the right, you can see the difference between, in one of those mākāhā channels, between high tide and low tide. So here, the mākāhā channel is almost filled with water, while on the lower picture, we have a low tide. Okay. So another component was to look at Fishpond Bathymetry. So we had a student measuring the bathymetry of the fish pond to get an idea. A really quick question? Or
yeah. You're measuring the water and the height. When the water comes in, does it thoroughly mix, or does the hot water stay on top or on the bottom?
This is, well, usually the warmer water would stay. There's 2 factors basically defining the layering of water in a water column. And it's based on density. One is temperature, so warmer water is lighter and will stay on top. And another one is salinity. And there's where it becomes really complex in the fish pond because you have temperature, differences, but you also got salinity differences because you got more saline water coming in from the ocean and fresh water coming in from the stream. So the fresh water will usually salinity is a more defining factor in determining which water mass sits on top. And so the fresh water from the stream will usually create a freshwater lens on top. I will have some salinity graphs later on where this is easily visible. So we collected fishpond volume, and because the height of, or the water level changes over time while we go from one station to another to collect water level, we also had a reference pressure deployed in the pond interior, so we could normalize the bathymetry data that was collected to one height. And we wanted to calculate, and here on the right you can see just a 3D plot of the fishpond bathymetry. So in general you can see it's really, really shallow, like less than half a meter in most places. And then in the northwest of the pond there's a little bit of a deeper area. And so we normalized our bathymetry data to 4 different tidal stages to basically, I want to go back to this one graph. We normalized it to these 4 tidal stages, so neap low tide, neap high tide, spring low tide, and spring high tide. And here you can see, and we did that in back in 2007, and then we recollected the data in 2018 because we were interested to see how the fishpond volume changed as a result of the restoration that has been going on. And so here you can see the difference in fishpond volume. So ocean break repair really results in a considerable change of fishpond volume from 2007 to 2018. And the addition of kahaokele, so of a new mākāhā channel, causes greater and faster outflux during spring, just because there's another channel that allows water to flow out, and also more influx during neap high tide. So that leads to a lower fishpond volume in 2018 during spring low and neap low, and to a higher fishpond volume in 2018 compared to 2007 during neap high, just because of this additional mākāhā channel that allows more influx or outflux. Spring high is the only non-expected value, basically. So we see higher fishpond volume in 2007 during spring high than in 2018. And we think that this might be because if I go to the next graph, you can see that this is how it looked in 2007. And at spring high tide, the wall was completely flooded. So it was basically at this tidal stage almost unconfined because water could really flow over the wall in this very large section. And that led to a greater volume than in 2007 for this tidal stage. This is on the right how it looks now. And so we also calculated water exchange, and we can see that water exchange increased, which is what we expected because we added a new mākāhā channel. 7% during spring tide from 2007 to 2018. And during ebb flood transition for neap tide, it increased 18%. And so the increase in water exchange really leads to a decrease in residence time from originally 48 hours in 2007 to 36 hours in 2018. So we also collected water samples. On the right, you can see the stations that we basically sampled. So we went with a boat. We had these interior stations that we collected, and it was really important to us also to go from a transect from one of those freshwater sources to the new mākāhā channel to really see how does the geochemistry change along this transect. And then we also sampled all the different mākāhā channels. And we had 2 end members. So end members means we had one pure ocean water sample and one pure here stream water sample, just so we could know how the geochemistry looks like in these 2 end member stations. And along with water sampling, we also used the YSI probe that measures water depth, pH, temperature, salinity, dissolved oxygen, and chlorophyll A. We measured those parameters at the water surface and at the bottom of the water column, because like you pointed out, the water can be really stratified due to density differences. There can be some layering, so we wanted to understand how does the whole water column look like. And here's just some pictures of us collecting water samples. There's Aka. He's also a graduate student in the Department of Oceanography, measuring water with a YSI probe. And this is just the water samples that we collected that we then brought back to the lab on the same day to be processed. So this is some of the salinity data that we collected with the YSI probe. This is the top, so at the water surface, salinity data from 2014, which represents before the repair of ocean break, and then 2017 after the repair of ocean break. And you can see that, first of all, salinity is a really good indicator of the relative importance of these 2 different distinct water masses in the fish pond and how they distribute. And so you can see that generally the northwestern portion of the pond, which is much more influenced by fresh water than the eastern portion of the front. And that's because here stream flows into the fish pond over here, while this eastern portion is connected to Kaneohe Bay. So this gradient really tells us how are the water masses distributed. And you can also see the difference from 2014 to 2017 in salinity. So we have much more freshwater influence in 2017. And we think that this might be because they removed mangroves around this section of the wall. And so the freshwater can flow much more freely into the fish pond. And this is the same sampling dates, just the bottom locations. And you can see that there's a less steep gradient in the bottom because the fresh water usually sits on top. So the bottom is actually much less influenced by fresh water. But otherwise, we can see pretty much the same pattern, a stronger gradient in 2017 compared to 2014. So now I'm going to go into the last point, which is what is the effect of circulation change on microbial communities. So our hypothesis was we didn't really know what to expect, but for sure that circulation, because the physics and the biology are usually closely interlinked, that circulation affects abundance of microbial indicators. And so this is just a depiction of, like, basically when we talk about microbial indicators, these can be different bacteria, and usually the source of bacteria can be fecal contamination, usually from humans or different animals that come into the environment via streams or oceans, and then they can survive in the environment, and if exposed to these bacteria, that can potentially lead to infection or disease. And so microbial source tracking quantifies the presence of microbes that live in the same conditions as these pathogens, but are present in much higher concentrations, and usually indicate usually representative for the proportions of these pathogens in the water. So we use these indicator bacteria to get an idea of what's in the water and what it could be associated to, basically. And the target bacteria that we used, or the target indicator bacteria, are Bacteriodales, Enterococcus, and which are both bacteria that can come from humans and animals. And we also used Fusobacteria, which is a very bird-specific bacteria, and that was important to us because there's a mangrove island in the fishpond interior that hosts a large cattle egret colony that, if you have a closer look at the mangroves, is really full of bird feces. And so we hypothesized that this could be a large source of contamination to the fish pond, and we were interested in really looking at bacteria specific to these birds and how much influence it has on the water quality. And then we can link this pathogen risk to fish pond circulation. So we took those water samples back to the lab, and they were processed for the analysis of nutrients and also for the analysis of DNR to be able to quantify the abundance and diversity of bacterial composition in the fish pond. And we are using the 16S gene that is basically conserved in all bacteria, so you can use it to ID bacteria in different ways. And this is just a picture of me processing some of the samples and preparing it for qPCR, which is a technique to amplify bacteria that then gives us data on which bacteria are present and how much of them are present. So this is some of the data of the Enterococcus indicator bacteria. And you can see on the left, these are the sampling dates in 2014, which is again representative for pre-ocean break repair. And on the right, you can see post-ocean break repair dates. And you can see that there's a slight increase in Enterococcus indicator bacteria over time. And the red line down here indicates the EPA maximum quote, which is basically set by the Environmental Protection Agency. And we hypothesize that the increase in Enterococcus bacteria is due to an increase of freshwater, because these bacteria are much more likely to survive in freshwater. So this could be one of the possible explanations of why we see a slight increase. And this is a similar plot for Bacteriodales. It's also slightly higher after restoration.
Could it also be due to circulation before the restoration?
We don't think so, because the circulation actually increased due to the addition of this new mākāhā channel. So before restoration, the whole wall was overgrown by mangroves, and there wasn't a lot of water flux in and out. So so we are pretty sure that the circulation actually increased. So it must be, it's more likely that it is really this additional fresh water coming into the pond that causes very favorable conditions for bacteria to survive. So Bacteriodales also slightly increased. So, and then the last one, which is the bird's fecal indicator, actually decreased slightly. And we think that this might be because it's not a bacteria that is so dependent on the fresh water. So we think then compared to Bacteriodales and Enterococcus. So in this case, we think that it actually could be the increase in water exchange and circulation that led to the decrease. Because probably before, they are now also restoring the Mangrove Island and cutting it down, it probably was way worse even before they started the Mangrove Island removal. But we definitely want to do some further research to make more, to be able to draw more conclusions from this first data that we collected. So my main conclusions are that water exchange is driven by tidal forcing and stream input, and that the addition of this new mākāhā channel resulted in a considerable increase of water exchange and therefore in a decrease of water residence time from 2007, so before the ocean break, to 2018 after the ocean break repair. And that placing an additional mākāhā in the seawall really improved circulation in the southeastern portion of the fish pond. And it looks like Ha'aukele accounts now for a significant share of water volume flux when we compare flow at these different mākāhās. And lastly, the changes in circulation that do seem to have an impact on microbial distribution in the pond. And we would like to do some more analysis to really figure out what causes the pattern that we see. Some implications of this work is that fish kills may be avoided in fish pans if the fish pans are moved to strategically favorable regions for fish survival. So we have had some major fish kills in the pond before, and that was likely because they were placed in locations in the pond where there wasn't a lot of water exchange. So now we know where water exchange is large, and this could be a strategically good place to place these fish pans. And understanding the physical environment in here really helps us to advance our knowledge of dynamic biochemical and physical interactions in Hawaiian estuarine ecosystems. And generally, those results can also help us to evaluate and guide future restoration steps. So yeah, with this, I would like to conclude. These are my resources. And I don't know, do we have a little bit extra time to show the video?
We got about another 17 minutes.
Okay, perfect. So in that case, I would like to show you a video that really depicts the essence of the work that Paula Moehlenkamp does over there. Oh, did I? Yeah.
Can you say how many acres are in this pond?
88.
80?
88. It's pretty large. Yeah.
Is the Mangrove Island a natural feature?
No. No. I will explain after how it developed. Yeah.
Is there a long-term intention for this to work and be sustainable?
Yes.
Versus research to apply to fish ponds also.
Right.
But this one.
We're standing here at a fish pond. This is an 800-year-old fish pond built for the community of Hawai'i. And I think community is a big part of what we're trying to establish here, is that fish ponds are made for people, not just families. We sit in a crisis right now where food production in Hawaii is not what we're striving for, right? And I think that's the ultimate thing we should strive for, is to be producing things for ourselves instead of relying on somewhere else in the world to feed us. Today was a community work day. We had about 80 volunteers out here working for the restoration of this pond. The foundation of this pond is built upon the people who come out and volunteer here. As a staff, we could not do what we do without the help of community. We're at a fish pond. We like to talk about fish and where does fish lie within each and every one of our daily life. It's our food source, yeah. 800 years ago is their main protein. Today, life is a little easier with stores. Yeah? We can drive to the store. We can go to Costco. We can even order things online and it can come right to our doorsteps. And that's where we actually miss a connection with our environment. We need to look to our cell phone to tell us what's going on in the world instead of being out in the environment and telling us what's happening by itself. And I think today the people who actually came out here and put their hands to work connected with their environment a little more than they had been previously. It's awesome to support the cause. It's awesome to be an advocate for good work, but things don't get done unless you come out here and actually get your hands dirty. And in Hawaiian, there's a great saying, it's huli kalima ilalo eola. That's when you turn your hands down to the soil and you live. Because the only way something can grow, the only way you can feed yourselves is by getting your hands dirty. And today, each and every one of our volunteers got their hands very dirty. I know the awesome thing is you can see what happened today. You can look back on the day and see what you accomplished. And whether you come back the next day, the next week, the next month, or the next year, progress is going to continue to happen here at Fishpond because volunteers and community, they come together and they really support what we're trying to do. And the ultimate goal is to feed people. And as an island sitting in the middle of the Pacific Ocean, we should have no problem feeding ourselves fish. And yet, right now, we rely on 80% of the rest of the world to feed us. That's fish is coming from somewhere else constantly. I think one thing people need to take away when they visit these places is no matter where they live around this island, no matter where they live around this world, they have an effect to the ocean. Whether it's through your storm drains, whether it's littering, out of your car, an accident, or on purpose, we all have slight effects on this ocean. Little do we know that this ocean provides us with oxygen, with food, with just a place to be able to go, and spend time with our families, and how important this ocean is to each and every one of us. Even though we might live way up on the mountainside, this ocean is important. Little steps like coming out and volunteering at places like Fishpond. This is not the only place to volunteer. Get out in your own communities. Do good works around your own household. Make a garden at your house. Those are all small steps to making community stronger. And once we can build a stronger community, then we don't have to rely on the rest of the world to feed us, to clothe us, to get us through our daily lives. That's what this island is for. 800 years ago, they're fully sustained upon what they produce. Even up to 100 years ago, Hawaiians fully sustained themselves on this island. And I think, still with some good work and some good help, we can still sustain ourselves today.
So yeah, I thought this really shows very well, because it shows what Hypotheses does and what their mission is at the fish pond. Yeah, so any questions?
What's the average depth of water in the pond at low tide?
At low tide, very, very low. So maybe less than half a meter. Yeah, so maybe 30 to 40 centimeters. That's one of the reasons we had to make use of kayaks then, because the boat couldn't go anymore.
Any control, or maybe it's already controlled by the state or whatever, but of pesticides, insecticides upstream? I saw bacteria, but I didn't see chemical analysis.
Right. So yeah, there's another project further upstream where they are working on restoring the low E. So this is really, as I explained earlier, a very integrative system where things are interlinked. So when we work on the fish pond, it's also really important to make sure that upstream we do the work of putting these lo'i in place that can capture the sediment runoff. I don't know too much about if they do pesticide measurements up there. I would have to look into it, but they definitely look at things like water quality as well. So I would imagine they might. Rosie, maybe you know more than me.
Yeah, so they do, the state of, the Department of Health does measure certain pesticides, like they measure Trojan, I believe, and a couple of other pesticides that have historical use. Those data can be found online, I believe, on the Department of Health database. I don't know how frequently they sample, but they do sample, like semi-infrequently. Yeah,
and about the Mangrove Island, the question that you asked, so it was actually naturally created in a way, although the mangroves were invasive and that were intentionally introduced. But what happened during the 1965 flood is that part of the wall got broken and washed into the pond. And that was then where the mangroves started growing on top of these wall leftovers. Yeah.
So you're raising some fish there. You have two places. Are those fish donated to the public? Do you have a big luau once in a while, or do you sell them?
So I think right now the fish is not commercially sold, but what they do have every now and then is like fundraisers where they invite people to come and work. I believe they just recently had one, and then they catch some fish and make it for the people that came to the community work day. And
so in 2009, there was a really dramatic fish kill where about 10,000 fish died. Prior to that, they had grown the fish in fish pens and sold it to market. But because of the tragedy that happened that pretty much wiped out all of their fish stock, they realized that they had to redirect their efforts from fish production to restoration to making the environment much better for them to grow the fish in. And so since that time, since 2009, they really focused on the restoration so that they can grow the fish in a much better, they just realized that the environment wasn't as suitable. They are growing fish for experimental purposes in terms of, asking the question, like, is the restoration working? Is it helping them to grow fish? And Paula's right, they do sell it at their fundraisers, but they also have a small amount that go to local restaurants. So there's gorilla ogo that they harvest that goes to some local restaurants, and also salmon crab. Other products, so not necessarily like fish, but other kinds of seafood are sold to specialty restaurants.
Talking about pesticides before, I seem to remember years ago there were a couple of universities involved with testing all the way from water there and water upstream. And you said those are published?
Those are not published in terms of papers, but they're publicly available. I know online the Department of Health publishes some of that data. And I know that there are a couple of researchers at the University of Hawaii, Henrietta Delay, who does test a couple of specific pesticides.
Are there any cesspools upstream?
Yes, Haiku Plantations is completely on cesspool and septic tank, despite the fact that all of those residents could probably afford to be on sewage line. Yeah, and so that there is an effect. You can see pulses during storms of
Quick question, can the decrease in the Fusma bacteria post-restoration be because of the clearing of the mangrove along portions of the habitat for the cattle egret?
Well, interestingly, we see the cattle egret only on the island. No one of us knows. We already tried to connect to people that are more experts with birds. But our observations, and this also aligns with what Paipai'o here observes, is that those cattle egrets mostly stay on the island. And one reason could be that the predator pressure is very low over there because it's in the pond interior. But we don't know for sure. But yeah, there's only cattle egrets on this island. Yeah.
So what was the purpose of the island?
Well, the island was created naturally due to this. So the mangroves just started growing on the resident on the leftovers of this pond wall that was washed into the interior. And now the island is actually being completely, I believe they finished their work now. So over the past couple of months, they have been completely clearing the island from mangroves, and they are planning to plant native species there.
What was when they showed the construction of the new wall being put in, and you could see rocks on either side?
Right.
What were these rolled up yellow forms that were built down into it?
I believe, is it concrete? I think it's concrete that they sometimes use as well.
Tubes, not tubes, but
Yeah.
cylinders of concrete?
Yeah, exactly. Originally, so back in the days, they only used the basalt rocks and the fossilized corals that are pretty porous and allows a little bit water exchange as well. And now they utilize some of these concrete cylinders.
Like zero exchange, near zero from the wall versus the channels or the...
Yeah, I think there's still through pores some exchange, but it's very difficult to quantify.
Yeah.
So we limit our quantification to the mākāhā channels. Yeah.
Have there been any major wall failures since it was rebuilt?
I'm sorry?
Have there been any major wall failures since it was rebuilt?
No. But we do see now at the king tides that we had a couple of months ago that they come very high and start overflowing the wall as well. And there was a question.
Do you have any plans to remove the sediment that was from the mangroves in there?
That's a really good question. I don't know. I think that would be beneficial potentially because, these sediments do take up a lot of space, but I'm not sure if there's right now any concrete plans in place to do that.
Historically, did the Hawaiians actually move the sediment back up into the valley? Or did they handle that?
Well, historically, there wasn't that much sediment at the fish pond because those low-E and the native vegetation captured most of the sediment runoff. So this is really a problem that was created as a result of land use change in the last century.
Is there cattle back in the watershed?
I don't know. That's a good, is there?
There were cattle, like way back, like maybe 100 years ago.
Yeah.
And then there was pineapple and then rice. There are in the Hawaiian newspapers accounts of the fish pond caretakers tying like coconut fronds to the end of a ahupua'a or a canoe, and then on the outgoing tide, swiping, kind of cleaning the fish pond that way. But clearly it was not as sedimented as it is now, and so the pond would probably be a little bit deeper if we got rid of it.
Yeah, so true.